Okay, so today we are going to start a new subject. We have been essentially looking at how to cut genes and paste them, etc. And we haven't really looked at how genes are actually controlled. It's kind of useful to have some control on gene expression, right? You wouldn't want all your 21,000 genes in your body being expressed all the time in all the cells, right? It will be kind of a mess. And also in bacteria, you need to have some kind of control on the gene expression so that you're not wasting precious material. So most organisms have developed, well, all organisms have developed some way of controlling expression of the genes. That means, so if you have some genes, only some of them may be being expressed at any one time. So we understand some, to some degree, the control of gene expression, but not all of it, especially in eukaryotic cells. Our understanding of gene expression actually started with bacteria. So some time back, if you read the book, you'll see some time back two people named Jacob and Monod. They studied E. coli and expression of an enzyme called beta-galactosidase and some associated proteins with it. And they found out that these bacteria have a pretty elegant mechanism for controlling the gene expression. So, for example, well, let me go back. So, for example, if we have E. coli bacteria growing in a medium, and that medium does not have any lactose in it, well, then the E. coli doesn't need to be producing any enzyme that breaks down the lactose. Remember, lactose is a sugar which is a disaccharide. It is made up of glucose and galactose. So in order to be able to digest that, we need to have an enzyme that breaks this down, and then the bacteria can utilize the glucose for its purposes. But you need the enzyme to break it down. Now, if there is no lactose in the media, then the E. coli doesn't need to produce beta-galactosidase, the enzyme, to break this down, right? So there is a control mechanism that stops the production of beta-galactosidase if there is no lactose in the media. On the other hand, if there is lactose in the media, then the cell would like to break it down and utilize it, right? So in that case, the presence of lactose actually turns the gene on, and that's the way it is controlled. So we are going to look at this mechanism of how gene expression is turned on in this particular case. So gene expression is the overall process of information flow from genes to proteins. We know that already, right? And the way it is controlled, well, there are several levels of control in eukaryotic cells, but mainly it is controlled at the level of transcription. So remember, we have the DNA, and here is the gene, and here is the promoter. So so the RNA polymerase is going to come and bind to the promoter. Okay, and then it goes down the gene, reads the gene, and makes the messenger RNA. Right? Makes the messenger RNA. So Uh, the control of gene expression is in either preventing the RNA polymerase from binding to the promoter or in somehow preventing it from going forward. Okay, so there are various mechanisms that different organisms have developed. Depending on the gene and the organism, there are different mechanisms how this is controlled. Okay, but the basic idea is to either prevent the RNA polymerase from binding to the promoter or to make it difficult for the RNA polymerase to go forward, right? And if you want to, so that's called negative regulation. And another type of regulation is if you make it easier for the RNA polymerase to go forward to read the gene. That's called positive regulation. So there are two types of regulation: negative regulation and positive regulation. Okay, we'll talk about that in a minute. All right, so a gene that is turned on is being transcribed, is being actively transcribed. That means it is producing messenger RNA that then can be translated to protein. Okay. So different organisms respond to the environmental changes by controlling the gene expression. So it may turn the gene off. So when we talk about gene being turned off, that means the expression, it's not being transcribed. Okay, and when we say gene is turned on, it means it is being transcribed. Okay, so in bacteria, in eukaryotic cells, prokaryotic cells, so this applies to prokaryotic cells only, there are things called operons. So an operon is a group of genes under coordinated control. So for example, the gene that I was talking about, beta-galactosidase gene, it is part of an operon called the lac operon. So let me draw a picture of that. So for example, here is one gene. This is the beta-galactosidase gene. Okay, and then there is another gene which is called the permease gene. So this is the permease gene. Sorry, the handwriting is pretty bad here. And the third gene after that. So this is a cluster of three genes, which is controlled by a promoter which is upstream of the first gene. So here is the promoter. Well, in fact, in this case, there is a promoter, but there is also an operator. So let's call it. So here is something called an operator. And this area here is called the promoter. Now, I just want to make sure everybody understands. These are all this whole thing is nothing but DNA, right? Sequences of DNA. So the beta-galactosidase gene is a particular sequence of DNA. The permease gene is another sequence of DNA, and the third gene, I forget the name of that gene, is another sequence of DNA. Same with the operator, it's a sequence of DNA, and the promoter, another sequence of DNA. So this is nothing but ATGC sequences. Okay. Now, what happens is, so this whole thing here, these three genes, the structural genes, and the operator and the promoter, this whole thing is called the operon. Okay, and in this case, because it is controlling the beta-galactosidase gene, it is called the lac operon. Lac operon. So there are many different types of operons in bacteria, in prokaryotic cells. Okay, so so in the case of the lac operon, there are 3 adjacent genes for lactose utilization enzymes. So the beta-galactosidase gene is actually the one that breaks down lactose. Permease genes it makes the protein that allows lactose to enter the bacteria. Okay, so there are 3 different enzymes in a row, and the promoter sequence and the operator sequence. Okay, so this whole thing is the lac operon, and the way the expression of these genes is controlled is by interaction of these 2 elements, the promoter and the operator, with different proteins. So you see, DNA interacts with different proteins to control the expression of genes to make other proteins. Okay, so let's take a look at how that works. Well, it turns out there is another gene called the regulatory gene, which is upstream of the lac operon. So remember the lac operon, which are the operator, the promoter, the operator, the LAC-Z gene, the permease gene, and the third gene. This is the lac operon, and this is the long piece of DNA. This is on the chromosome in the bacterium. And somewhere around here, there is another gene called the regulatory gene. And this regulatory gene codes for a protein called the repressor protein. Is everybody with me so far? Okay. All right. So now, what does this repressor protein do? Well, these are just E. coli bacteria. Okay. So what does the repressor protein do? Okay. So here is a better picture than what I was drawing. All right. So remember our 3 genes right here. The beta-galactosidase, the permease, and the third one right here. Okay, that's our lactose utilization genes. And here is the operator. Now, the repressor protein, here is the repressor protein that is made by the regulatory gene. So here is the regulatory gene making the repressor protein. Okay, the messenger RNA from the regulatory gene is right here. So the repressor protein will go and bind to the operator. So when it binds to the operator, our RNA polymerase cannot go and bind to the promoter. It's not allowed to go and bind to the promoter because this repressor protein is blocking it. And because it is blocking it, the gene is turned off. So the gene expression is turned off, right? So in this case, when the repressor protein is active, gene expression is off. Okay. And notice that they call it active repressor. That means it can bind to the operator. Well, it turns out that this protein interacts with lactose. If there is lactose present in the media, that lactose molecule goes and binds to the repressor and makes a conformational change that makes it inactive. That means it cannot bind to the operator anymore. Right? So when that happens, our RNA polymerase is free to now go and attach to the promoter and go down the cascade of genes, read the genes, and make the enzymes necessary for breaking down lactose. So guess what? So now these enzymes can go and attack the lactose, break it down, and the cell can utilize it, so utilize the sugar, right? So isn't that neat? When you don't have lactose, the repressor keeps the genes inactive by blocking the expression of the genes, and that saves energy for the cells because they don't need to be making all that protein, right? But as soon as there is lactose present, it blocks the repressor. It makes the repressor protein inactive, and all of a sudden the bacteria can turn on the machinery for making these enzymes that break down the lactose. So it's kind of a self-fulfilling thing. If there is lactose present, the gene expression is turned on. If there is no lactose present, the gene expression is turned off. So the bacterium is very efficient in utilizing the lactose this way. Okay, so this is called negative control. This is negative control because it works by either turning the, blocking the RNA polymerase from acting. Okay, negative control. Okay, so there is another type of negative control. Okay, so here are two types of operons. This is the lac operon that we just talked about, right? Okay, and this kind of operon is called repressible operon. Okay, hold on just a minute. Okay, so no, this type of operon right here, this is called the inducible operon. Inducible operon. And that's because when you have lactose, it induces the expression of the gene, right? So by putting in lactose, you are inducing the expression of the gene. That's why it's called inducible operon. Okay, and there is another operon called trip operon. This is called repressible operon. Why do we call it repressible operon? Because in this case, if tryptophan is present, then the gene expression is repressed. The gene expression is repressed. So, and when tryptophan is not present, the gene expression is turned on. The reason is the trip operon controls the genes, the set of genes that help the cells make the enzymes that produces tryptophan, right? So if there is no tryptophan in the media, then tryptophan is an amino acid that's necessary for cell growth, right? So if there is no tryptophan in the media, then the cell will want to make tryptophan. And so you want the enzymes to be, the genes to be transcribed, right? But if you give the cells tryptophan in the media, then they don't need to make their own tryptophan. And so then the tryptophan represses the the operon by making the repressor active. Have I confused you? So they act in kind of similar way, but in one case, presence of lactose induces the gene expression, and in the other case, presence of tryptophan represses the gene expression. Okay, because in one case, the cell needs to break down lactose, and in another case, the cell needs to make tryptophan for itself. Okay, so these are two different types of operons, inducible and repressible. Both are negative control. Negative control. Both are examples of negative control because you stop the RNA polymerase from binding and reading the gene. Okay. Let me make sure everybody is on the same page. Everybody is understanding what I'm saying. Okay. Victoria says yes. Okay. So okay, so Kathy is saying that seems like when the ribosome subunit got blocked. Yeah, it's kind of similar. You're basically blocking the expression of the gene. You're blocking the RNA polymerase from reading the gene, okay, from going down the gene, reading it to make the messenger RNA. So if you can't make the messenger RNA, then you can't make the protein. So that's essentially it, okay. So far we have discussed negative control, negatively controlled operons, and within the negative control you have inducible operons and repressible operons. Okay. All right. Now we also should talk about positive control. Positive regulation, positive regulation. So negative regulation and positive regulation. So what is positive regulation? Positive regulation is when you help instead of blocking the RNA polymerase, you actually make it easier for the RNA polymerase to read the gene. So the lac operon has both negative and positive regulation. Okay. How is that? So when there is lactose present, then the lac operon is turned on, right? So the operator is not blocked. Okay. But what if you have lactose present in the media and you also have glucose present in the media, if you also give glucose to the cells? Well, if there is glucose in the media, then the cells do not want to utilize lactose because glucose is a better carbon source for the cells, right? So in that case, even though the operator is not blocked, the RNA polymerase that binds over here doesn't transcribe the gene efficiently. But if glucose is used up, and you have lactose, then what happens is the cell makes a protein called cyclic AMP. And that cyclic AMP binds near the promoter and makes it easier for the RNA polymerase to go down the gene. So it kind of helps the RNA polymerase. So that's why we call it positive regulation. So cyclic AMP helps the RNA polymerase move down the gene. So So the presence or absence of lactose is negative regulation, and the presence or absence of glucose makes for positive regulation. Okay. Okay. So the best possible scenario for lactose utilization enzymes to be produced is when when lactose is present. So what happens when lactose is present? The repressor is made inactive, right? The repressor is inactivated. And the second condition is glucose is absent. Does that make sense? If glucose is absent, then cyclic AMP is made, and it helps the RNA polymerase transcribe the genes better. Lactose is present, it makes the repressor inactive and frees up the operator. Okay, and glucose is absent, makes cyclic AMP and makes the transcription more efficient. Okay, so that's why this is negative regulation, and this is positive regulation. Okay. Any questions so far? Okay, so these are just two examples of bacterial operons. There are many different types of operons in bacteria, in prokaryotes, and they work kind of by similar principles. So there is either negative regulation or positive regulation. There is always a promoter to which the RNA polymerase binds. There may or may not be an operator, and then depending on the mode of action, the RNA polymerase is either prevented or helped. Prevented from reading the enzyme or helped in reading the enzyme. And that's how genes are regulated in prokaryotes. Now, there are some promoters that are called constitutive promoters. Constitutive promoters, they are always on. They're always on. That means there is no control. So there are some proteins that the cell needs to make no matter what, right? So for those proteins, it doesn't need to have any regulation. And so those are called constitutive promoters. So if you want the protein to be produced constantly, you would use a constitutive promoter. Okay. And then there are some promoters that are inducible. We just saw an example of the inducible promoter. The lac operon is an inducible promoter because you need lactose to induce the promoter, right? If you don't have lactose, then the gene is turned off, and if you have lactose, then the gene is turned on, right? This is kind of useful. Having inducible promoter is kind of useful. So think about our when we are constructing plasmids. So if we have a plasmid with our gene of interest, over here, and if we were to make a promoter, upstream of it, if we were to have a promoter, if we have to have a promoter upstream of it, then it would be nice if this promoter were inducible. Why is that nice? Because you could first grow the cells without inducing the promoter. That means you are growing the cells without making any protein, right? And you can grow the cells to high density. So first, you grow the cells to high density. So in this phase of cell growth, we are just focusing on making the cells grow very fast and to very high density, right? Once you have cells that have been grown to really, really high density, then you can just add the inducer. And what's going to happen now is all of a sudden, all the plasmids in all the cells are going to be turned on. The genes are going to be turned on, and all the cells now start to make lots and lots of proteins. Okay, so this is a very efficient way of making proteins in bacteria. So in most cases, in most companies, they use inducible promoter of some sort that is used, and so they grow up the cells to very high cell density. That's called the growth phase, and then they will induce the gene expression by adding something or changing certain conditions. For example, there are some promoters that are temperature sensitive. So you can grow the cells at 37 degrees, and then you raise the temperature to 42 or 40 degrees, and all of a sudden the gene expression is turned on, and your cells start to make lots of protein. There are other inducers that use chemical. There are other promoters that use chemical inducers, of course, and some promoters are pH sensitive. So there are many ways to control gene expression in bacteria, and it is done very commonly in fermentation when you are growing bacteria to produce the desired protein, right? Recombinant bacteria. So constitutive and inducible promoters. Most of the time we use inducible promoters in biotech companies. Okay. All right, now let's switch over to eukaryotic cells. Eukaryotic cells, we don't have operons, but the expression of genes is controlled in similar manner as the bacteria, but it's a lot more complex. It's a lot more complex. So let's first think about how the eukaryotic genes are packed inside the nucleus. Remember, in eukaryotic cells, the DNA is all inside the nucleus. Have you heard of the word chromatin? Chromatin. So inside the cells, inside the nucleus, there is DNA, but in eukaryotic cells, the DNA is complexed with proteins. So there is a complex of DNA and protein, and that is called chromatin. Okay, there are certain types of proteins that it is complexed with. Okay, so so let's take a look at how the DNA is packed inside. So here's the DNA molecule. Long. Remember, it's very, very long, right? In eukaryotic cells. So here's the DNA molecule, and the way it is packed is it forms these loops. Around protein molecules called histones. So these purple molecules are protein molecules called histones. So it's almost like a string of beads, right? So here is one bead, here is another bead, another bead. These beads are made of histone molecules, and the DNA is wound around it. Okay, so that's the first level of packing, right? That's the first level of packing. So you are forming a coil or a string with the DNA. Well, you have the DNA string, and then you're forming a coil with it, with these beads of histones. Now, that coil is forming a rope by a second level of winding. See that? The second level of winding right here? That's a second level of binding. So this is called the tight helical fiber. and then that tight helical fiber winds around like a rope. Okay, see this rope winding? That's the supercoil. So you can see how tightly the DNA is packed in a chromosome. So here is our chromosome right here. Okay, and within that, all this DNA is packed tightly. And there's lots of protein in there and DNA. And because of this tight packing, so you can imagine if your gene is somewhere in there, if your gene is somewhere inside, the RNA polymerase may not have access to it, right, because of the tight packing. So because the genes are tightly packed, RNA polymerase normally doesn't get access. Polymerase access is blocked, right? It's physically blocked. So this is kind of the first level of control of gene expression in eukaryotic cells, the DNA packing. So you literally have to somehow unpack the DNA to be able to get to the gene, right? So DNA packing is one level of control in eukaryotic cells. Well, also each gene has its own promoter and terminator region. Okay, promoter region and again, promoter is where the RNA polymerase binds. Okay, but the gene expression is usually switched off, and they require activators to be turned on. They require activators. What are activators? These are again proteins. So gene expression is often controlled by lots of different proteins. Okay, so activators are types of proteins that interact with the DNA. Okay, and it's not just one activator. In this case, it's controlled by interaction between lots and lots of regulatory proteins and control sequences. So what do we mean by control sequences? Those are DNA sequences on the DNA itself that may be either next to the gene that's being controlled or a little bit farther away too. Okay, so control sequences we are referring to DNA sequences, and regulatory proteins are of course proteins that are produced by the cell. Okay. Okay, so what kinds of regulatory proteins are we talking about? So there are things called transcription factors. Transcription factors, they promote RNA polymerase binding to the promoter. So they help the RNA polymerase to come and bind to the promoter. Okay, and then there are other activator proteins that will bind to the enhancers. Now what are enhancers? These are DNA sequences. Okay, these are DNA sequences that are, we'll talk about, I'll show them to you in a picture soon so you will understand. And then there are other proteins called silencers that can repress or inhibit the transcription. Okay, so there are many different ways in which gene expression is accomplished in eukaryotic cells. Okay, so in this case we talked about two control sequences. The promoter is where the RNA polymerase binds. Okay, so promoter, and the enhancer. Enhancers are where the activator proteins bind. Okay, so let's take a look at a picture here. All right, so here is my gene. Here is the gene. Okay, and as usual we have the promoter over here. Promoter is nothing but a sequence of DNA, right? And I have two sequences over here called enhancers. Again, these enhancers are not close to the promoter. They are farther away, right? And so you can see all these proteins. Some of them, these are called transcription factors. Transcription factors can include activator proteins. Activator proteins are the ones that bind to the enhancers. Okay, And then they in turn bind with other transcription factors. And what it does is basically make a bridge over here so that the DNA molecule loops around and makes this nice spot for the RNA polymerase to bind. You see how that works? So it requires all these different proteins to make gene expression happen. So what if one of these proteins is absent? Then either the gene expression won't happen or it will be less efficient, right? So you can see how you can literally control the expression of genes by the presence or absence of any of these proteins. Presence or absence of any of these proteins. So if you didn't have these activator proteins, then of course you won't have gene expression because this looping of the DNA won't happen and the RNA polymerase won't be able to come and bind here, right? So this is one way of how gene expression is controlled in eukaryotic cells. Any questions about this? Okay, so Victoria is asking to repeat what happens if other proteins are absent. So think about these, see what all these proteins are doing. All these proteins are forming a complex. They're forming a complex. So it's like building a bridge, right? And if you don't have one of these proteins, if this protein is not there, then this bridge either won't form or it will form, it will be less, it won't have the right shape, right? And then the binding of this RNA polymerase either won't happen or will be less efficient. Yeah, so that's why. And then Kathy is asking if the protein is missing and you wish to activate the cell, it can be manually added. Yeah, could you add the protein? It's difficult to get proteins into the cells. The cell has to make its own protein. But many times there are signals, small molecule signals that you can add that will trigger the production of a particular protein in the cell. I'll give you an example of that towards the end of this lecture, okay? But yeah, so you can actually control the production of some of these proteins by signaling. Okay, so you could have some control over gene expression that way, and your body does have control over that, and it actually turns the production of some of these proteins on and off depending on the conditions. Okay, this is how the body controls gene expression in many cases. Okay, so this is a picture from the book, and it gives you a little more detail of how the promoter acts, the promoter. So here is the gene, and here is the promoter. This is the promoter. Now, the promoter has, of course, many types of sequences. And one of the important type of sequence is called the Tata box. It's called the Tata box. So this is a DNA sequence. And why is it called the Tata box? Because, of course, it has the sequence, Tata, and then several A's after that. This is the consensus sequence of the Tata box. Okay. So why is the Tata box important? The Tata box is important because it will bind to Can you guys hear me now? Okay, good. Sorry about that. I don't know what happened. All right. So we were talking. I'm going to clear the page. Okay. So we were talking about this Tata box binding protein. So this is just one of the many proteins that are around. So you have Tata box binding protein and then other associated factor. All these things form a complex called TF2D. Okay, and they come and bind to the Tata box, and then other proteins come along and form a certain shape, and because of that certain shape, the RNA polymerase can come and bind to the promoter. Okay, so this whole thing is called this is the promoter, and the Tata box is a particular sequence in the promoter region. Okay. Now that is not the only sequence in the promoter region. There are several other sequences. So if you read the book, they will talk about the CCAT box or the CG box. So there are many other boxes around that have different functions. They will attract different proteins, different transcription factors. So what happens is these transcription factors will come in and bind to specific sequences over here, and they will associate with the this complex here, the TF2D complex. And by that association, they all have their specific shapes, and so they have specific attachments, and they form. Basically, you're forming the right shape for the RNA polymerase to come and bind to the DNA, right? So you can see how many different proteins are around. What I'm trying to show you here is that there are certain sequences on the promoter, like the Tata box and the CG box and the CCAT box. Those are all sequences that have specific affinity for certain proteins. And because of that, the proteins are attracted to that, and you form this nice complex of proteins that allows the RNA polymerase to bind. By the way, you notice this has a name called Pol2. So there are different types of RNA polymerases. There is Pol1, Pol2, and Pol3. Now, Pol2 is the polymerase that is involved in messenger RNA production. Okay. Whereas Pol1 and Pol3 may be involved in other types of RNA, like ribosomal RNA production. Okay, so most of the time when we are talking about messenger RNA, we are talking about Pol2, the RNA polymerase. Okay, so this is how complexes are formed in eukaryotic cells. So another example of that. So this is similar to what we saw before in the color picture. Here is an enhancer sequence, and here is your gene sequence, and here is your promoter. Sorry, the whole thing is promoter. The gene sequence is beyond this. Okay, so so there is an initiation factor which will come in and bind to the promoter, and an activator protein will come and bind to the enhancer region. And then remember the loop forming, and the loop forms by the association of the activator protein with the initiation factor, this association right here. And because of that, this specific shape is formed, and your RNA polymerase can then come and bind to that shape and start to read the gene, right? So the thing to point out here is that not all control sequences are right next to the gene. The promoter is right next to the gene. Here is the gene, and the promoter is right here. But this control sequence here, the enhancer, is far away from the promoter, right? But because of this looping that is formed, even though it is far away, you can bring them close to each other, and it's actually beneficial for the RNA polymerase to bind, right, to make that particular shape. So enhancers are sequences of DNA. Those are control sequences that are usually far away from the promoter. But nonetheless, they will bind with the activator and form a loop that will help the transcription process. Okay. So in general, these activator proteins, they bind to the DNA with one surface. They bind to the DNA with one surface, and they bind to other proteins with the other surface. You get that? So the shape of these activator proteins is really important. They bind with one surface to the DNA and bind to other proteins, the other transcription factors, with the other surface. Okay. All right. So, Kathy, you were asking a question about controlling. Can you put in a protein to make the thing happen, make the transcription happen, right? So there we have a gene for a protein called beta interferon. Beta interferon. So this beta interferon is useful in fighting viral infections. Okay. So if you don't have viral infection, then you don't need the beta interferon in great quantities, right? But when you have viral infection, then you want to produce, you want your body to produce a lot of beta interferon. So guess what happens? When our body gets infected by a virus, it induces the production of some of these proteins. It induces the production of some of these proteins. And by doing that, now it makes it possible for the, see how all these proteins, the HMG and the JUN and the ATF and the NF kappa beta B, etc., they are all coming together in a specific way to bend the DNA molecule so that RNA polymerase can come in and bind to it and then produce beta interferon, which can then fight the infection. So the viral infection actually triggers the cells. So it actually, the virus doesn't literally make the protein, but it triggers the production of certain proteins that will then enhance the expression of the beta interferon gene. Okay. So what I've tried to show you today is there are lots and lots of different ways of controlling gene expression at the transcription level, okay, at the transcription level. And it's quite complex. Not all genes are controlled the same way, right? You've seen many different ways in which proteins interact with the DNA to control the expression. And I've shown you a few ways in which that can happen, right? Questions so far? Questions? So Kathy is asking, if you have an immune issue, does that mean that interferon cannot be made by your body? I don't know if I know the answer to that. I don't know what you mean by immune issue. That means your body is not doesn't have the immunity. Oh, like HIV. All right, HIV doesn't. I don't think it interferes with the beta interferon production, but I think it acts in. It integrates itself into your chromosomes, and when the conditions are favorable to HIV, it will come out of the chromosome and make lots of virus. And another problem with the HIV is that it keeps mutating, so our immune response doesn't work all the time. So HIV is a retrovirus, so it can make it has an RNA in it. It has RNA in it, and when it comes in, it makes DNA, and the DNA gets into our chromosome. All right, so let's review what we've learned. So can you describe to me what are the different elements of a bacterial operon and their functions? Give me 3 different elements of the bacterial operon. Okay, Victoria says 3 adjacent genes. Well, that's for the lac operon. It doesn't necessarily have to be 3 genes, but it's a cluster of genes, okay? So, cluster of genes. Yeah, that's one. That's one element. What's next to the cluster of genes? Yeah, there's the operator sequence, correct? Operator and then the promoter. Okay, so that's the operon. Sometimes there may be a terminator region at the end. Bad handwriting. Okay, so give examples of a negatively and positively controlled operon. I should say negatively and positively regulated operon. So a lac gene, lac operon. Kathy says it's negatively regulated. Yes, it is negatively regulated, but it is also positively regulated, right? We saw both examples in the lac operon. So we had negative regulation by lactose. Okay, and the same operon is positively regulated regulation by glucose. It's a little confusing because it doesn't mean that if you have glucose, it will produce the gene. It actually is the other way. If you don't have glucose, it will produce the gene, but it does so by helping the RNA polymerase. That's why we call it positive regulation. And the lactose, when it is absent, it blocks the RNA polymerase. That's why we call it negative regulation. Okay, so whenever an operon is blocked by something, then it's negative regulation. And when it is helped, when RNA polymerase is helped by something, then it's positive regulation. Okay. So examples are both lac operon is an example of both the negative regulation as well as positive regulation. Okay. All right. What's the difference between a constitutive and an inducible promoter? Yes, constitutive is always on, and inducible is turned on when an inducer is present, right? is turned on when an inducer. Now, inducer can be a chemical or it can be a condition. So you could change the temperature and induce a promoter, right? All right. How does DNA packing control gene expression in eukaryotic cells? Yeah, no access, no physically no access to the gene for the RNA polymerase, right? Yeah. So Kathy says makes it difficult for RNA messenger. It's not the RNA messenger; it's the RNA polymerase, right? To access the gene, the promoter. Okay. What is a Tata box? And what is its function? So the Tata box is a sequence of DNA with the consensus sequence, with this consensus sequence. Now remember, the Tata box doesn't always have this sequence. There may be some variations in that. Okay, but and this sequence is in the promoter region. And what is its function? Its function is to bind to a particular protein called the Tata binding protein, and that starts the process of the initiation complex. The initiation complex for transcription. So it's a really important sequence where the initiation complex is formed, okay, by binding to the Tata binding protein. Okay. And what are enhancers and how do they interact with transcription factors? So are enhancers proteins or DNA sequences? That says protein. That's not true. So so remember, enhancers are sequences of DNA that are further away from the they are further away from the promoter region. Okay, but they interact with activator proteins. They interact with activators. Activators are proteins, right? So they interact with activators, and then the activators bind with the other transcription factors that will form the loop. That allows the RNA polymerase to bind to the promoter, right? So is that clear? Enhancers are DNA sequences that are away from the promoter, and they bind with activator proteins, and the activators then bind with other transcription factors to form the loop. Remember that loop, the DNA loop that allowed the RNA polymerase to come in there. Okay, so I think that's it. What questions do you have about today's lecture? Okay, Victoria's question is, will today's lecture be the quiz for next Monday? So this coming Monday is a holiday, right? So February 18th is a holiday. So we won't have a quiz that day. And so February 25th, we will have today's lecture and next week's lecture. Okay. Okay, so Kathy has a question. When you grow cells to high density, is that the same as the log phase of the curve we looked at last week? All right, so let's take a look at that. So when you're growing cells, so here is the cell concentration, and here is your time. So what I was talking about in E. coli, what we want to do is you want to keep growing the cells. You start with the lag phase, remember? And then this is the exponential growth phase. And then they start to slow down over here. This is the log phase. And this is the stationary phase. Now, this is very high cell density, right? Right here. So when you are producing a protein, what you want to do is you want to get them through the log phase all the way up to stationary phase, right? You have made lots and lots of cells. And now when you're entering stationary phase, that's where you want to induce them. That's where you want to induce them. So now, after this, the cells are not growing, but they are just producing lots and lots of proteins, right? And you may want to feed them some media that you need for protein production, right? So you are literally taking the cells through the log phase, and just as they are about to enter the stationary phase, when you have grown the cells as much as you can, right? Then you start the induction. Then you induce them, and then you just allow them to make protein for a while. And what happens is in the bacterium, so here is the bacterium, and once you induce them, you will start making so much protein because the cells are not growing anymore, they are just making protein. So the whole cell becomes packed with these inclusion bodies that are just protein, denatured protein. So those are inclusion bodies. Now this is protein that you later on you will break open the cell and you will take this protein out, solubilize it, and then you will refold it to get your and then you'll purify it, to get the protein. But this is how it's done. It's basically you keep growing the cells until they cannot grow anymore. You've basically taken them to the highest density possible, and then induce them. Okay, other questions? Okay, so Kathy is asking, so if you're trying to increase the efficiency like we looked at last week, that happens at the mid phase. But oh, okay, so the efficiency that we talked about last week was efficiency of transformation, right? So how do we get plasmids into the cells? Okay, so um, so that's very different from what we are discussing today. So here, when we are growing cells, those are already transformed cells. Okay, once you transform the cells, now you can freeze them, and then you can grow them for production. Okay, so what we talked about last time was when you want to transform cells. Okay, so you have a cell that doesn't have any plasmid, so you want to grow it and you want to capture it in the log phase, and that will allow the plasmids to go in. So plasmid is the DNA, right? Once the plasmids go in, now you can freeze it, and now you have these cells that are recombinant cells, and you can grow them. So when you grow them for production, that's what I'm talking about here. Okay, so two different things. Okay. Any other questions? If not, I will see you online next Wednesday, right? Online next Wednesday. And then in the lab on the 25th. Okay. So I will stop the recording.